It's such a privilege to be back here at London Calling. This is an event that means a lot to me. For several years, Oxford Nanopore Technologies was my home. And at this event last year, I hosted the Diagnostics Day, where we showcased how this technology is being taken into multiple clinical applications globally. And it was great to hear an update from Gordon this morning about just how many of those clinical applications are progressing. And it was really that ambition about taking this groundbreaking science into clinical impact that's led to my current role at the Ellison Institute of Technology in Oxford. And I'm excited to share a little bit more about what we're working on with all of you this afternoon. So, as Aaron mentioned, I started my career as a medical doctor. I practiced in the NHS for a number of years, and it was really during that time that I came to appreciate how important multidisciplinary team working is across systems in order to achieve collaboration and drive real impacts for patient care. So that's really what the collaboration is about between Ellison Institute of Technology and Oxford Nanopore Technologies. And I am guessing that the Ellison Institute may be new for some of you because it's only been around about a year. So I thought it might be worth just introducing a little bit more about what we're seeking to do at the Institute and then talking about infectious disease. So this is really the vision of Larry Ellison. He is the co-founder of Oracle, and Professor Sir John Bell, who for many years was the Regius Professor of Medicine at Oxford University. He's now the founding president of EIT, and during and since the pandemic, both Larry and Sir John have spent years, in fact, discussing this vision for what they want to do with EIT, which is about developing and deploying science to solve real-world problems. So what you can see behind me is a picture of the Oxford campus, which is being built at scale and pace over the next couple of years. It's been designed by Sir Norman Foster. And it's really designed to bring together those multidisciplinary teams that I was talking about. In this case, it's teams with expertise in engineering, in AI, scientists, and entrepreneurs. And it just happens to be over the road from Oxford Nanopore Technologies, meaning we can work closely with teams there. So we have a bold and ambitious agenda for the campus, a bold and ambitious agenda for the problems that we are seeking to solve through this institute. And there are a number of programs being stood up in parallel. One is the Generative Biology Institute. We've been joined just this week by Professor Jason Chin from Cambridge. He is going to be pushing frontiers of synthetic biology and precision medicine. There's another big investment being made in the field of artificial intelligence, which includes building a health foundation model, bringing together DNA sequences, medical images, clinical information, really with the goal of improving our understanding of human health. We're also launching a program in food security and a scholars program, which speaks to the investment and commitment to not just build teams today, but to build teams that have skills needed for the future. So my role is as Chief Executive of the Pathogena program. This is the first scientific program to be launched at EIT. A number of the team are here today as well. And our program is being stood up to tackle infectious disease, bringing metagenomic next-generation sequencing into point-of-care diagnostics. And the reason why we're doing this is to get an ongoing data feed from devices to create a global genomic infrastructure that's going to improve patient care and give real-time information at a population level about what infectious disease outbreaks are happening where, really with the overarching goal of preventing the next pandemic. And that's really where this program was born out of, the last pandemic, a moment when I know lots of people in the room stepped forward, and part of the challenge at that time was to collate information from multiple labs, tests, scientific teams globally about what was happening. That was the genesis for the vision from both Larry Ellison and Sir John Bell to have a real focus on what it would take at a system level to prevent the next pandemic. They realized that it would take more than just investing in a specific test or advancing a specific piece of technology, but really needs an investment in global genomic infrastructure and at a whole system thinking level. One of the ways to think about the Pathogen program is that we're building the digital microbiology lab of the future. And with that in mind, we're working closely with colleagues at Oracle. This is enabling us to build out the infrastructure at a global scale from the outset and to do that in a secure setting. The reason why this all matters, as everybody knows, is that there is a substantial probability of another pandemic occurring in the next decade. We also know that antimicrobial resistance is rising. Gordon spoke about it this morning. Millions of people are dying because of antimicrobial resistance. So this is where the features of Oxford Nanopore's technology really differentiate themselves with regards to the rapid turnaround times, the accessibility of the platform, all make it a critical tool in this fight. So our goal at EIT is to advance the field of metagenomics, to improve clinical decision-making, and to tackle AMR to make a difference to people's lives and improve outcomes. To do this, a number of things need to happen, which I'll walk through a little bit about embedding this technology into health systems, feeding it back to clinical teams. This is why the link with the EHR is critical. So what I wanted to set out here is how we're thinking about it in the EIT Pathogena program. So what you can see on the bottom of this schematic is the global data platform. In year one of the program here, and I look forward to updating where we get to with this, the platform is based on Oracle infrastructure, and it has information about thousands of different pathogens that can cause human illness. But not just the information about the pathogen, it's got clinical metadata and where we can has accompanying MIC data. We need this in order to inform clinical teams about what bug they're treating and what the sensitivity is to make the best prescribing decisions. To date, we've got over 5 million sequences in the platform. We know we need to go much further. What you can also see in the top of this schematic is how the point-of-care diagnostic that we're working on fits in. We will also have data-driven support systems that are enabled by machine learning and artificial intelligence. And all of this collectively is what creates the always-on monitoring system that we're here to talk about. This gives us an early warning system of disease. Today, what we have in EIT Pathogena is a couple of bioinformatics platforms. These are available and being used by customers globally for SARS-CoV-2 and mycobacteria. These are cloud-based. The MicroTool is fully automated and gives predictive antimicrobial susceptibility testing across a wide range of drugs. The data is stored in the cloud, and these tools have been validated. Having these tools out there today as we build the platform and work on the diagnostic are enabling us to get real-time feedback from public health agencies globally as we further develop what devices and products are going to be of future impacts. So, as most of you know, over the last couple of decades, the Oxford Nanopore team and platform has really redefined what's possible in portable near-to-real-time genomic sequencing. And when I joined EIT in September last year, one of the first things we had the opportunity to do is to set up a collaboration between EIT and ONT that would really leverage some of the capabilities of the platform, and that's what I wanted to share an update with you today. This is using Oxford Nanopore's Mark 1D device as part of a global initiative. And this initiative is about providing real-world performance data from a diverse range of environments, as well as getting that data flow securely from ONT devices directly into the data platform in the cloud that we're building. We need to do that in a secure way that is consistent with all data governance requirements in different countries. This is why it's a global collaboration built on the future clinical utility of the Nanopore device. So I'm delighted to share where we are with the global collaboration today. The blue dots that you can see on the map are where we are deploying Mark 1D devices as part of this fully funded 12-month initiative. A lot of the teams on both ONT and EIT have worked incredibly hard to get these sites ready and signed up. And it's important to call out, these are not traditional Nanopore customers. These are people that wouldn't necessarily be using Nanopore before, and the purple dots represent some of our other collaborations. This is deliberately global from the outset because we're trying to solve a global problem here, and so we need that diverse geographical spread. This is really about getting genomics out of the lab into real-world clinical settings. So we're just getting started with what we're working on at the Ellison Institute. And I wanted to close by thanking both the EIT and ONT team for their support in setting up this really exciting collaboration. We look forward to continuing to work with all of you about using this technology, in this case specifically about transforming the field of infectious disease. So thank you for the opportunity.